Logo for NIH, National Eye Institute. Program Overview, National Eye Institute. Translational Research Program, February 11, 2022. Tony D. Gover, Ph.D., Program Director, Bioengineering and Technology and Cornea Injury and Repair. Tony.Gover@nih.gov.
Good morning, everyone. I'm Tony Gover. I'm the Program Director for Bioengineering and Technology and our Cornea Injury and Repair program. So today I will be providing a brief overview of NEI's Translational Research Program, and specifically the R24 mechanism released by NEI. So there are many funding and resource opportunities for translational research within NEI and the NIH.
Slide, NEI Translational Opportunities.
Listed on this slide are some of the main opportunities for translational funding available to NEI researchers.
NEI Translational Research Program, TRP, on therapy for visual disorders.
So in addition to the translational research program I will talk about today, the NEI specific opportunities include our small business program that provides early stage capital for innovative small U.S. companies, and our Audacious Goals initiative that is focused on regenerative medicine. Now, the AGI program does not have a specific focus on translational research. Its mission is actually much broader, but it does provide certain opportunities for translational funding. There are also a number of collaborative programs at the NIH that includes the Office of the Director, together with multiple NIH institutes and centers that support translational research. These would include the NIH Blueprint program that is focused on accelerating transformative discoveries in brain function in health, aging, and disease. And within the Blueprint program, there's the Blueprint MedTech program that is focused on translating medical devices. And then there's the Blueprint Neurotherapeutics program that is focused on translating biological and small molecular drug-based therapeutics. Now, one of the greatest advantages to working with these Blueprint programs is that in addition to the funding we can provide directly to the researchers, the funded researchers also have the opportunity to collaborate with NIH-funded consultants and can augment their project with NIH contract research organizations that specialize in things like regulatory policy, manufacturing, toxicology, and clinical testing. And this is at no extra cost to the researcher. So this program is particularly useful for products with, say, complicated manufacturing processes or a complicated regulatory pathway, or for investigators or groups that just have less experience with these types of activities. The Brain and Heal initiatives.
NIH Brain Research through Advancing Innovative Neurotechnologies, or BRAIN Initiative. NIH Helping to End Addiction Long-Term, or HEAL Initiative.
Are two other NIH-wide programs that, although not focused specifically on translational research, do provide certain translational opportunities in their areas of brain health and pain and opioid management. Finally, NEI works very closely with our partners at the National Institute of Biomedical Imaging and Bioengineering on encouraging bioengineering solutions for diagnostics and therapeutics by releasing multiple funding opportunity announcements. So you can find more information on all these translational opportunities on our website,
www.nei.gov/grants-and-training/funding-opportunities.
But I would highly encourage anyone interested in these programs to discuss these opportunities with your program officer. These programs have very different missions and requirements, so you want to make sure your applications are aligned to the most appropriate program possible.
Slide, NEI Translational Research Program, R24.
So the NEI Translational Research Program,
TRP,
is focused on translating innovative laboratory research findings into therapies for use by clinicians to treat visual systems diseases or disorders. And these therapies may be biologics, small molecular drugs, devices, or combination therapies. Now, we execute the Translational Research Program by releasing an R24 Funding Opportunity Announcement, or FOA.
Number PAR-20-319.
And the focus of the application should be on obtaining the preclinical data necessary to support an IND or IDE application to the FDA. Now, We understand that there's an incredible amount of data that is required to submit an IND or IDE application, particularly for new regulated entities. And so the funding ceiling is very high for these awards. You may request up to $1.5 million per year in direct costs for up to 5 years. Additionally, since the application should be focused on translational research specifically, the scientific and technical merit review is conducted by a special scientific review group convened by the NEI for this very purpose.
Slide, NEI TRP Investments FY06-21.
So we have actually had the R24 mechanism for many years, but prior to 2006, the R24 translational program was really focused more on creating infrastructure and core facilities for translational resources rather than a focus on a particular regulated product. So today, I will just be sharing information from 2006 to the present. And even from 2006, the R24 FOA has changed over the years, requiring more and more mature products as an entry point to the program, and a shift to products that have a clear medical indication rather than platform technologies. Okay, so since 2006, 20 awards have been made at an average cost of just under $9 million per award.
Total investment was $175,368,286.
Nearly half, 45% of these awards are still active. And so we should keep this in mind as we go through the next few data slides.
R24 status, 45% or 9, active totaling $69.8 million, 55% or 11, closed totaling $105.5 million. Therapy type.
Not surprisingly, the vast majority of awards are focused on biologics.
15.
With only 4 awards focused on small molecular drugs and only 1 device. And I will talk about that device in a few slides. Even though we only have 20 awards in this portfolio, the portfolio is very diverse. These awards are addressing over 13 different diseases, including diseases of both the anterior and posterior eye segments.
NEI TRP Award Primary Disease Focus, achromatopsia, age-related macular degeneration, autosomal dominant retinitis pigmentosa, cone dystrophies, diabetic retinopathy, dry eye disease, glaucoma, Leber's hereditary optic neuropathy, or LHON, limbal stem cell deficiency, retinitis pigmentosa, retinopathy of prematurity, Stargardt disease, vitelliform macular dystrophy.
So it is really great to see that our translational research program may have a great impact for so many diseases, and more importantly, on so many people's lives.
Slide, NEI TRP Award Productivity.
Okay, our translational research program investigators have been very productive. So far, they have over 850 publications and at least 15 patents. And it should be noted that many of these investigators had secured their IP prior to their R24 award. So these patents are not included in that 15. And I will share one such project with you in a couple of slides. But this is actually a very good thing because secured IP, of course, significantly decreases the overall risk to product development. So far, our investigators have 8 granted INDs or IDEs for their original primary indications. And there are several products that now have additional approvals for other indications. And I will show you at least one example of this as well. There are now 7 active clinical trials, again, for the original primary indication, and we know of several more that will soon be initiated. And finally, we have 1 product associated with FDA market approval. And remember, 45% of our translational research awards are still active. So we expect these numbers to increase significantly over the next few years. So over the next 3 slides, I thought I would give you an example of 1 device, 1 small molecular drug, and 1 biologic that continues to progress towards the marketplace.
Slide, transscleral drug delivery for retinal disorders.
So first we have our single device.
R24EY017045.
This original effort was led by Dr. Henry Edelhauser from Emory. And the focus of this R24 was to explore and develop novel transscleral drug delivery approaches to deliver drugs to the retina and other tissues at the back of the eye.
using nanoparticles, microneedles, fibrin sealant, ontophoresis, and electroporation. The PI proposed to A. Examine the physiology of transscleral drug transport, B. Establish analytical methods to determine the distribution of low molecular weight drugs as well as macromolecules to the various regions of the posterior segment, C. Demonstrate delivery and therapeutic effectiveness in experimental animal models following transscleral drug delivery, D. Develop the technique of drug delivery to the suprachoroidal space following a single microneedle injection. A diagram of a cross-section of an eyeball shows that the suprachoroidal space, a layer surrounding the eyeball, is occluded anteriorly by the scleral spur. Another diagram of an eyeball shows that suprachoroidally injected fluid spreads circumferentially between the sclera and the choroid around the globe.
In one approach, the team studied injection into the subchoroidal space, the space between the sclera and the choroid tissue layers, using a microneedle. The free length of these microneedles is only 900 microns, so the needles are very, very tiny. The suprachoroidal injection technique allows clinicians to target therapies to retinal tissues, potentially maximize efficacy, while compartmentalizing therapies away from the unaffected vitreous and interior segments. This technique also allows clinicians to potentially deliver retinal therapeutics in an in-office based setting.
Product development status. Developed a minimally invasive delivery method using hollow microneedles that can locally target the suprachoroidal space, SCS, for drug delivery to the back of the eye. 79 published articles, 3 patents, 7918814, method for drug delivery to ocular tissue using microneedle, 109055586, methods and devices for drug delivery to ocular tissue using microneedle, 8197435, methods and devices for drug delivery to ocular tissue using microneedle.
So to further develop and market this device, the PIs of this award started the company Clearside Biomedical. Their first FDA-approved product for suprachoroidal administration is Xipere. Xipere is a proprietary suspension of a corticosteroid, and it's indicated for the treatment of macular edema associated with uveitis.
This is the first FDA-approved product for suprachoroidal administration.
And as you can see from the slide, the company and their collaborators are pursuing several other drug combinations and indications with this microneedle device. This includes therapies for AMD, diabetic retinopathy, ocular oncology, and other retinal diseases.
Slide. Photo-switchable channel blockers for treatment of blindness. R24EY023937.
The second effort was focused on a small molecular approaches to restore vision for potentially numerous degenerative diseases. This effort was led by Dr. Russell Van Gelder from the University of Washington. The team focused on restoring light perception to patients with photoreceptor degeneration by making the retinal ganglion cells, or bipolar cells, photosensitive with the use of small molecular photoswitches. So photoswitches are pharmacological antagonists or channel blockers that use an azobenzene, a light isomerizable chemical moiety, to block voltage potassium channels.
The PI proposed to test photoswitch compounds in vitro and in vivo in mouse, rat, dog, and primate models to determine which to take forward into clinical development. Proposed an in-depth analysis of their effects on the primate retina in vitro and proposed to conduct the required preclinical toxicology and efficacy tests with the goal of applying for a new drug application to allow human clinical studies at the conclusion of the award.
So, on the far left of this slide, we see an example of one of the photoswitches the team developed. In the presence of light, the compound is in the higher energy cis form. In this form, the compound can block potassium channels of the retinal ganglion cells or bipolar cells, depolarizing these cells and driving them to generate an action potential, making these cells light sensitive. In the dark, the photoswitch relaxes back to the trans form, allowing potassium to flow and hyperpolarizing the neurons.
Structure of BENAQ is pictured. Visible light converts BENAQ from the trans to the cis form, and then the compound quickly relaxes back to trans in the dark. A schematic depicts use of ligand-gated non-selective cation channel to load membrane-impermanent photoswitches into cells.
So the IP for this technology, which was secured prior to the R24 award, was recently licensed to a company for further clinical development. Now, Kiora Pharmaceuticals is planning clinical trials for this year with one of these molecular photo switches. And it is for patients with late-stage retinitis pigmentosa.
Lab developed second and third generation photoswitch compounds and successfully applied the small molecule photoswitches to large animal retinas, demonstrating efficacy in several retinal degenerate models. 16 articles were published. Intellectual property for the photoswitches was successfully licensed by UC Berkeley to Bayon Pharmaceuticals. Bayon Pharmaceuticals has been subsequently acquired by Kiora Pharmaceuticals. Kiora Pharmaceuticals plans to initiate a Phase 1b clinical trial for KIO-301 photoswitch compound in the third quarter of 2022 in patients with latter-stage retinitis pigmentosa. Schematic showing modes of photoregulating of potassium channels, one at 380 nanometers, one at 500 nanometers or delta. Slide RAAV-CNGB3, Gene Therapy for Achromatopsia, Translational Research Studies, R24EY022023.
Okay, the third and last effort I will highlight is a biological therapeutic for achromatopsia. Achromatopsia is a rare autosomal recessive cone disorder with an estimated prevalence of 1 in 30,000. It typically manifests within the first few weeks or months of life, and affected individuals suffer from nystagmus, photophobia, severely reduced visual acuity, and color blindness. Now, 5 genes have been implicated in this disease, with a mutation in CNGB3 accounting for nearly 60% of those with the disease. And this gene encodes for the beta subunit of the cyclic nucleotide gated channel expressed in cone photoreceptors. So this effort was led by Dr. William Alsworth from the University of Florida. And this translational research award was focused on developing a recombinant adeno-associated virus gene delivery reagent for human CNGB3.
The PI proposed a set of coordinated activities aimed at enabling the translation of a successful preclinical gene therapy for the CNGB3 form of achromatopsia into a clinical trial. This novel recombinant adeno-associated virus, RAAV, gene delivery reagent is a first-in-class therapeutic agent to address the unmet vision needs of patients with CNGB3-related achromatopsia. AIM-1 will optimize the RAAV-CNGB3 vector by systematically modifying each of the components, promoter, intron, cDNA, and poly-A. AIM-2 will produce RAAV human CNGB3 vectors for use in animal studies. AIM-3 will evaluate an alternative serotype, capsids, and cone promoters for efficiency in a CNGB3 negative mouse model of achromatopsia. AIM-4 will evaluate CNGB3 achromatopsia patients using a battery of state-of-the-art methods to establish natural history, appropriate inclusion-exclusion criteria, and endpoints appropriate for the clinical trial. AIM-5 will use the optimal vector from AIM-2 to perform GLP-compliant toxicology and biodistribution studies in rats and non-human primates. AIM-6 will prepare and submit an IND to the FDA. Graphic of AOSLO images from 2 subjects with ACHM. 4 squares progress from darker texture to lighter texture.
So as a result of this award, the investigator was able to submit an IND application to the FDA, which was granted. And currently, Applied Genetic Technologies Corp., who was a collaborator in the original award, is conducting a multi-site phase 1-2 clinical study to evaluate the safety and efficacy of this genetic therapeutic.
A multicenter clinical trial for CNGB3 achromatopsia gene therapy employing an AAV vector carrying the normal human CNGB3 gene, cDNA, was approved by the U.S. FDA. 18 articles were published. Ongoing Phase 1-2 study of the safety and efficacy of AGTC-401, administered by subretinal injection in individuals with achromatopsia, caused by mutations in the CNGB3 gene. The primary study endpoint will be safety, and the secondary study endpoint will be efficacy. Slide, takeaways.
Okay, and so the takeaways. There are numerous translational opportunities available for NEI investigators. And we, the NEI, plans to continue to support translating therapeutics to the clinic and the marketplace.
The NEI continues to support innovative and productive researchers to translate medical products to the clinic. Investigators should reach out to their program officers to determine the best translational opportunity.
I would highly encourage applicants to reach out to us early in the process to discuss what opportunities and resources are available to help accelerate the development of potential therapeutics. And finally, please take advantage of these funding resources. I think one of the greatest threats to all of our translational programs is the lack of competitive applications. We actually receive very few applications for all of the programs I mentioned in the beginning of this talk. So the resources are out there, so please take advantage of them. And with that, thank you for listening, and I'm happy to take any questions.
Logo for NIH, National Eye Institute.